we have seen in detail how a chromosome is formed. At the same time, we did take into consideration certain aspects which were important for the formation of chromosomes, and we saw it from that point of view, how the chromosome is functioning when the cell division is taking place, when a cell develops, what are the changes that a chromosome undergoes, how a chromosome is working, how a chromosome is arranged. So, if I have to surmise the entire thing that you have studied while you studied the structure of chromosomes, we can say that it is an arrangement of chromatin material, right? You know very well what is chromatin now, and while you were reading about the chromosomes, you had been introduced to the terms heterochromatin and euchromatin, and this would be what we have to study in this very short lesson, where you will get to know what is heterochromatin and what is euchromatin in a eukaryotic chromosome. Only the points of differences will make you understand what it is. You know where it is located. It is the chromatin material that surrounds the nucleosomes. Along with the nucleosomes, you have that highly coiled material forming the chromosome. Now, if we have to observe that chromatin material which takes up the color in a chromosome, it could be divided into two categories, one being heterochromatin, other being euchromatin. What does word eu means? Eu means true. So, that chromatin material which would be true, that means this would be false. It is not the case. Euchromatin refers to that chromatin material which is genetically active. That means rest of it is inactive. Is it lazy? When I say genetically active, it means that that part of the chromosome which is going to contain the genetic information that actually gets expressed. Now, while you study further, you will get to know that out of all the information in DNA, not everything has to be processed and not everything becomes concrete in the form of proteins. For now, just remember the points. You will, analyze the statement in great depths, and you can associate, once your knowledge about genetic processes is complete, then you can make it out that what does this genetically active and non-active means. So, instead of using genetically active, when I make you learn the differences, you are going to remember that in place of genetically, I am going to use the term transcriptionally active. Now, what transcriptionally active means? That part of the chromosome that undergoes transcription. We will come to know what it is. Now, we have heterochromatin and we have euchromatin. This makes as much as 97% of the chromosomes, and this is as less as 3%. This one is transcriptionally active. Pardon me, it's not, this one is not transcriptionally active, this is transcriptionally active. That means the true chromatin material is the material which will express itself, okay? The part of the chromosome that will express itself and its genetic information is going to get expressed, that is known as euchromatin, okay? Now, this heterochromatin is lightly stained. No. I am getting mistaken, pardon me for this. This heterochromatin is, first of all, you have to remember, it is tightly packed, while the euchromatin is loosely packed. This you can remember like this. Supposedly you are shifting to some place, or you have packed the material in such a way that you keep those stuffs which you require in the journey, or first of all, you keep those things easy, handy, okay, and those which are not required, you will keep those down where they will not be handy in their availability. So, in the same way, the one which has to give the information that has to undergo transcription is loosely packed so that it is easily accessible. Okay, the one part of chromosome that has the information in it, it is loosely packed. Instead of super coiling, over and over coiling, that part is loosely packed so that the information can be taken out without getting into much of the mazes that are created. But the one which has no information in it, that will be tightly packed. Alright, you are getting my point. What I am trying to tell you, like supposedly you have packaged your material, you are shifting somewhere, so you will keep those packages which would be required at the earliest at your ease, instead of packing them downwards very much in cluttered stage. So, you place the required things loosely so that you can access them easily. In the same way, the accessible part that is to be accessed is loosely packed as compared to the non-transcriptionally active part, which would be tightly packed, and it is of no use for the cell. It is not going to produce the proteins. Now, once which is tightly packed is going to take up the stain more. So, it is darkly stained. More would be the presence of nucleosomes, more stain they are going to take. So, this part is darkly stained, and because it is loosely packed, this is lightly stained. It takes up the stain in lesser quantity and it is lightly stained. So, you see within the chromosome also we are having a distinction of the chromatin material, where one happens to be heterochromatin and other happens to be euchromatin. This 3% of difference within the chromatin material makes us part of so much variety around the world. Alright, talk about human population only. You can just watch me and see yourself. There is a lot of difference, and where has that difference come from? It has come from this 3% only, out of which not all are expressed. That is also part of later studies, but this 3% is making you so different from me and others around us. Okay, so this 97% between you and me is similar because it is not expressed. And how does it matter whether it is similar or it is different? It is never going to be part of expression. Whatever information it has, it is not going to express. That is why it is not transcriptionally active. While which has the information, that is transcriptionally active. Because it has to be active, it is loosely packed. Because it is loosely packed, it takes up the stain lightly. So, these are the points that you have to remember in the case of heterochromatin and euchromatin while we talk about eukaryotic chromosomes. Alright. So, I hope you remember these things. You can associate with the structure of chromosome and identify it from this point of view, because further when we study about the genetic information being expressed, do keep in mind that it is out of that 3% only.